Hello everybody, welcome to In Vitro Science. Today I'll be talking to you guys about glycolysis. This is a very important topic. I think every science student out there, if they're doing science at university or even at standard A-level, this is something which you learn about. Of course, you learn about a bit more detail in university, which I'll be talking to you about today. So glycolysis, before I actually get into the main, diagrams of glycolysis, I'm just going to tell you what glycolysis actually is. Glycolysis is a anaerobic process, so it undergoes anaerobic respiration. Okay, so this means that you don't need oxygen. Okay, so you don't need oxygen, okay, for it to be carried out. So glycolysis is purely an anaerobic process. Oxygen is not needed. Glycolysis takes place in the cell, of course, and it takes place in the cytosol. The cytosol is the soluble compartment of the cell, sort of the aqueous compartment of the cell where organelles and the nucleus are suspended in it. So if I just show you a quick diagram of a cell, this is a typical eukaryotic cell, for example, with a nucleus. Let's say we've got some mitochondria in here. Let's say we've got, Golgi, for example. And the cytosol is basically, I'm going to draw it in yellow, is basically this yellow compartment here. So it doesn't actually include the nucleus, and it does not contain any organelles. So it just basically includes just the soluble compartment of the cell. And in the cytosol, you usually find things like the cytoskeleton, which is your microfilaments, which is your actin filaments, and your microtubules, and your intermediate filaments. Okay, so you find these things located in the cytosol. That's important. So the cytosol is basically what you have there. Okay, simple as that. Now there's a pretty important formula, not for you, just for you guys to remember, where you get glucose, and we know glucose is a 6-carbon sugar. And glucose essentially gets broken down into 2 molecules of pyruvate, or sometimes called pyruvic acid, but I like to call it pyruvate. And this is obviously a 3-carbon sugar. Because pyruvate is a 3-carbon molecule, of course 2 of those are required from one glucose. Okay, so glucose is a 6. So essentially here what we understand is that glucose is a 6-carbon sugar. And because glucose is a 6-carbon sugar, this one big molecule of glucose can be broken down into 2 smaller molecules of pyruvate. So you've got one big block of glucose, and that block of glucose can be broken down into 2 molecules of pyruvate. Okay, simple as that. What you also have to understand is where it takes place in. So as it's an anaerobic process, it takes place in all tissues. Okay, so every single tissues in the body, whether it being the brain, whether being the muscles, etc. This is where it takes place in. Everywhere, all the tissues in the body. So that's something for you not to forget. And also what's important about glycolysis is the main function of it is it makes energy. So you're trapping energy, but you're mainly synthesizing a very small amount of energy, where a net gain of 2 ATP, okay, molecules. And don't forget, your ATP is your adenosine triphosphate, and this is you're gaining basically net gain of 2 ATP synthesis. So this is your synthesis. Okay, so you're making 2 molecules of ATP. So glycolysis is also very important as well, because it can act as intermediates. What I mean by intermediate is that it can act as an intermediate. This is where things get a bit more important. Intermediate for fat synthesis. Okay, so it's a synthesis of fats, and also amino acid. Okay, so you can actually make fats, process known as the beta oxidation pathway. So from the glycolysis, you can enter the beta oxidation pathway to form fats, and you can enter the transamination pathway to form certain amino acids. So it's very interesting actually how glycolysis sort of branches off into different cycles, and that's the main thing. This is something which you can't cram. When I was in my first year of university, when I was so confused about what glycolysis is, I was trying to cram it. I was trying to look on YouTube for ways to memorize it, all the songs. I think that if you find it useful, then fair game, but I didn't find any of that useful. I had to sit down and actually understand what was happening. and then I understood it. So that's pretty awesome. So what I'm going to talk to you guys about now is glycolysis. Okay, so where do we get the glucose? So we know we start from D-glucose. Okay, so that's the first thing you have to know in glycolysis. We start from a D isomer or a D form of glucose, and D-glucose, as we know, is a naturally occurring most naturally occurring form is in its D form, so it's D isomer. That's why we call it D-glucose. And where do we get this D-glucose from? Well, we can get the glucose from sugars, of course, or we can get it from the starch, and starch obviously comes from plant material in your diet. Okay, from plants. Okay, in diet. We can get glucose as well from, let's say from breaking down of glycogen, glycogen in the liver. And we know that the breakdown of glycogen in the liver is a process known as glycogenolysis. Okay, so glycogenolysis. Lysis essentially means the splitting of glycogen to form glucose. Pretty interesting. We can also get glucose from things like recycled glucose. We can get it from lactic acid, amino acids, glycerol, etc., etc. But that's something we're not going to focus on now. But let's just these are the main 2 points I think you have to know about. So make sure you know those. But I'm going to talk to you about the cycle now, of where we're going now on the glycolysis pathway. So the first thing that happens is that D-glucose will form glucose 6-phosphate. Now, I'm not putting any of the drawings here because, I don't really have space to do that. But sometimes a lot of people find drawings helpful. So if you want to look at a drawing whilst I'm showing you this, then feel free to do that. But glucose goes into glucose 6-phosphate. Now, when I was at university, I was very confused about how did that take place? Well, it happens by an enzyme. Enzymes allow these things to take place. So it happens by a hexokinase enzyme, which is found in the muscle. But it can also happen by another form of enzyme, which is called a glucokinase enzyme. And that enzyme is found in the liver. So you find these enzymes which allow D-glucose to go into glucose 6-phosphate. And that happens by the aid of ATP. And what ATP does is that it will go in and ATP will sort of donate its phosphate to the D-glucose to form glucose 6-phosphate. So that phosphate molecule here is kind of being transferred onto that D-glucose molecule, which is pretty awesome because that's what things do. And something else for you to know, guys, if you spot these things now, it'll be very beneficial to you. You can see what I'm talking about. I'm talking about an enzyme which is called a kinase enzyme. And if you don't know this, a kinase enzyme is a type of enzyme which tends to phosphorylate things by using ATP. So in this case, we're using this energetically favorable reaction where we're using the energy to break down these phosphate bonds, these high energy phosphoanhydride bonds, to transfer one of the phosphate molecules from the ATP molecule to D-glucose to form glucose 6-phosphate. So that's pretty awesome. That's what happens. And then when we're going from glucose 6-phosphate, we can go from glucose 6-phosphate to another molecule called fructose 6-phosphate. So this is again another similar sort of structure to glucose 6-phosphate, but this time we have an equilibrium reaction. Okay, so and this reaction is these two molecules, glucose 6-phosphate and fructose 6-phosphate, these two molecules are isomers of each other. So these are actually isomers, structural isomers of each other, which means that you can rotate the molecules in a certain amount in space, so which is pretty awesome. Again, and another enzyme required to do this is something called phosphohexose isomerase. So phosphohexose, isomerase. Other textbooks say different things, by the way, for different enzymes, but they actually mean the same thing. So it doesn't really matter what enzyme you use. If you use my enzymes, I don't think you will lose marks on that. It's the exact same thing. So what we then do is that once we've formed our fructose 6-phosphate, we need another molecule of ATP in the system. So we have to get another kinase. Simple as that. Because we're forming another molecule now called fructose 1,6-bisphosphate. Now, another word I just want to bring out there to you again as well, guys, is just this word here called bisphosphate. No, I'm not using the word biphosphate. Biphosphate usually is used in science to represent molecules of two molecules. I'm using bisphosphate, and bisphosphate means two phosphate molecules. Okay, so that's what we use, not biphosphate. We use bisphosphate. Again, I'm not too sure on how your university works or how your school works. How the mark scheme works. You know, if you put bi instead of bis, you might lose. I don't know if you'll lose a mark or not. But anyway, so as you go from fructose 6-phosphate to fructose 1,6-bisphosphate, we're adding another phosphate group. Where are we getting that phosphate molecule from? Well, we're not going to get it from space because we're still in the cell. We're not going to get it from random places in the cell. We're going to get it from ATP. Don't forget that, guys. We are getting these things from ATP. ATP is crucial to provide us with that phosphate group. Very important phosphate group. We're getting that phosphate group from ATP to form our fructose 1,6-bisphosphate. Very, very important. Don't forget that, guys, please. And once we go to fructose 1,6-bisphosphate, we then have another enzyme called aldolase. And aldolase, essentially what it does is it's an enzyme, and it's a very naughty enzyme, I want to call it, because it kind of splits that molecule of fructose 1,6-bisphosphate into two molecules of this. So I'll just draw it first. So aldolase. So aldolase is an enzyme that will split fructose 1,6-bisphosphate into something called glyceraldehyde 3-phosphate. So we're splitting this molecule into glyceraldehyde 3-phosphate. Okay, so I'm just going to draw that right here. So glyceraldehyde 3-phosphate. Okay, and we are also going to split it to dihydroxyacetone phosphate. So dihydroxyacetone phosphate. Okay, and again, these two molecules are also reversible of each other. These are also sort of isomers of each other. Let me just do it a bit like that. So there we go. So these two molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, are also structural isomers of each other. Again, it can be rotated differently in space by the ratio of different molecules in different areas of the chain. And again, another enzyme is required, and this time this enzyme required in this case was triose phosphate isomerase. Okay, so it's called triose phosphate isomerase. Okay, so what do I need to tell you from here? So actually, sorry, I forgot to put this one in. So there's another enzyme here, phosphofructokinase. Okay, I just realized that now. So phosphofructokinase. Okay, so this is the enzyme which allows fructose 6-phosphate to go to fructose 1,6-bisphosphate, and then that can then split by aldolase into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, and those two molecules are in equilibrium because they're structural isomers by triose phosphate isomerase. And you can see that again, the word tells you everything. Isomer, they are isomers of each other. So don't just learn these things for the sake of it. Actually understanding this is going to prove great benefit to you guys. So now we're kind of done. Okay, so we're kind of halfway through the glycolysis cycle. We're not finished completely, but we are halfway through the glycolysis cycle. So if I just scroll down, guys, hopefully you've got everything there written down, or you screenshotted it, or you print screened it, or whatever you've done. So if I just scroll down now, guys, I just want to show you this. So we're kind of done with this molecule, so we don't really use this again. Okay, now we're going to focus on this one now. Okay, glyceraldehyde 3-phosphate. So what's going to happen now? So what happens here is that the glyceraldehyde 3-phosphate actually undergoes an oxidation reaction. And an oxidation reaction, what I mean by that is that it will undergo a reaction where it will also be in equilibrium. So if I just show you what I mean, you know, a lot of people are very confused. What does this guy mean? Well, this is what I mean. So again, this is also another equilibrium reaction, and we are going to 1,3-bisphosphoglycerate. Okay, I'm just going to draw the arrow a bit longer because I to ensure that I have enough space, and so you guys understand this thing. I don't want you guys not to understand that. So what happens then is we undergo a reaction where glyceraldehyde 3-phosphate can react with an enzyme called glyceraldehyde 3-phosphate dehydrogenase. So glyceraldehyde, 3-phosphate dehydrogenase. So what happens is that when it reacts with this enzyme called glyceraldehyde 3-phosphate dehydrogenase, it's kind of a long term, but if you just realize, we're dealing with G3AP, so glyceraldehyde 3-phosphate, we are going to just have to add dehydrogenase. So what does it mean if we're dehydrogenating something? We're kind of removing the hydrogen, So, and if you remember from I don't know if you learned this in high school or whatever, I learned there's something called oil rig. And oil rig is oxidation, is loss of electrons, loss of hydrogen, but a gain of oxygen. Now, I kind of modified this because in high school you probably learned oxidation is loss of electrons, full stop. And that's it. But what I modified it, I actually say, loss of hydrogen as well, because that's something for you to know as well. At university level, if you haven't learned already. Reduction is the gain of electrons, the gain of hydrogen, and the loss of oxygen. So in this case, we're dealing with an oxidation reaction, as I told you. So we're losing hydrogen, and that's why it comes from the word dehydrogenase. We're losing that hydrogen, to form a molecule known as 1,3-bisphosphoglycerate. 1,3, bisphosphoglycerate. So that's what's happening in this case. And what's happening here is we're directly adding a phosphate molecule. So we're directly adding a phosphate molecule in the system. So phosphate, I don't know how I'm going to draw this, but if you imagine this thing here, I'm directly adding an inorganic phosphate molecule into the system. But also, what's happening here is that you're having NAD+, going feeded in the system, going into NADH plus H+. And let me just actually draw that properly. So, cool. So we've done all of that. We've seen what's happening. But again, understanding is crucial. Glyceraldehyde 3-phosphate is being oxidized to 1,3-bisphosphoglycerate. So this is an oxidation reaction, But what's also going to happen, I'm just going to take that out because I tell you that's a bit weird. Your teachers might tell you that. But what's happening actually is that whilst glyceraldehyde 3-phosphate is being oxidized to 1,3-bisphosphoglycerate, your NAD+ is being reduced to NADH, Because reduction here, as we talked about, is the gain of hydrogen. And we can see when before we were deficient of hydrogen, and now we have that hydrogen molecule there. So it's kind of a redox reaction essentially taking place. So this is kind of a redox reaction, taking place because one molecule is getting oxidized whilst another molecule is getting reduced, which is pretty important because when you have, within the same reaction, when you have one becoming oxidized and one becoming reduced, that tends to cause a redox reaction. So don't forget that term, guys, redox reaction. So now we've got our 1,3-bisphosphoglycerate, but now again we're in equilibrium. So pretty much only about a few reactions in the glycolysis cycle is irreversible, about 2 or 3. So only 3 cycles actually are irreversible in the glycolysis cycle. So that the first few and then the last one. And whilst the rest are reversible, which is pretty cool because that means you can modify the cycle a lot. You can you can go the reverse of glycolysis by a process known as gluconeogenesis, where you can form glucose, but certain pathways can be overcome. But that I'm not going to go into detail with you guys at the moment. I just want you guys to understand the basics of what I'm talking about. So you've got your 1,3-bisphosphoglycerate. Now what you want to do is you want to form something called 3-phosphoglycerate. So 1,3-bisphosphoglycerate forms 3-phosphoglycerate. And what happens is when you're forming the 3-phosphoglycerate, this happens by a process known as substrate level phosphorylation. That means a phosphate group is directly transferred to ADP to form ATP. So this time we're not actually gonna, kill that ATP molecule. We're not actually going to use ATP. We're actually transferring this phosphate group from our 1,3-bisphosphoglycerate to our ADP molecule, so that can then go and form ATP. So that process is known as substrate level phosphorylation. Okay? So where direct transfer of a phosphate group is known as substrate level phosphorylation. So I want to talk to you about, guys, again, is an enzyme. Every step in the glycolysis cycle requires an enzyme, and this is no different for this step as well. And the enzyme which we require for this step is called 3-phosphoglycerate kinase. It's a type of an enzyme called a kinase enzyme. I'm going to put that in a different color, guys, for you. I don't want you to get confused, but it happens by an enzyme called 3-phosphoglycerate kinase. Okay, so 3-phosphoglycerate kinase. Again, a kinase enzyme involved in the ATP machinery, so it's kind of needed in the step. So then we go from 3-phosphoglycerate, we then go into another compound very similar but called 2-phosphoglycerate. And again, something for you to notice is that it is basically an isomer. Okay? And another enzyme is required, and this enzyme is called phosphoglycerate mutase. Okay, so phosphoglycerate or glycerate mutase. Okay, and then we're going from 2-phosphoglycerate, and we have another enzyme. So again, there's a ton of enzymes for you to remember, guys. But you will get it, don't worry. Just watch this video over and over again, and hopefully it'll get into your head. And this enzyme is called enolase. Okay, so enolase is another enzyme, and this time we're removing water. We don't need water, so we remove it out of the system, kind of dehydrating the compound, removing water, and that forms a compound known as phosphoenol pyruvate. Okay, and phosphoenol pyruvate can then go ahead and form pyruvate. And pyruvate is formed by an enzyme, very easy, called pyruvate kinase. And this is where ADP can then form ATP. Okay, so substrate level phosphorylation again. Okay, so it's kind of the same thing over and over again. Okay, so that's it, guys. Before I actually end this video, I want to speak to you guys about what I've just talked about briefly again. In terms of, there's actually 4 steps you have to know about. Okay, so there's an activation step. Okay, I'm going to do it in red. There's an activation step, not action, sorry. An activation step, and that uses ATP. So that's the first thing we did. We activated the whole system by using ATP. When we're going from this one, for example, this one, we use ATP to activate the steps. We then had another step where we split the 6-carbon sugar into half. Okay, so for example here, this is where we did it. We split fructose 1,6-bisphosphate into half. And from this point onwards, we're making 2 times of everything. So here, for example, we're starting with 1 times 6 carbon, 1 times 6 carbon, 1 times 6 carbon, 1 times 6 carbon. But as soon as we're splitting it, we're going to go in times 2. Yeah, so we're going in times 2. Times 2, with 2 everything, because 2 times 3 is 6. So 2 times 3 carbons is going to be 6 carbons. So again, everything from here, we're making 2 of everything, 2 of everything, 2 of everything. Okay, so we make 2 of everything, 2 pyruvate molecules from 1 glucose. That's why we get the term times 1, 6 carbon glucose, okay, forms times 2, 3 carbon pyruvate. That is what that term comes from. Okay, so you have to understand that. Again, not only that, times 2 of the enzymes as well. Times 2 of the ATP and ADPs. So don't forget that as well. Okay, so don't forget that as well, guys. Important. And then we talked about oxidation. So oxidation, again, we talked about oxidation from this molecule to this molecule, and then how we made ATP by substrate level phosphorylation. So not only using ATP in glycolysis, but actually places in the brain, for example, where we don't have, we can't have any, oxygen, no, where we can't, places in the body, organs in the body where, strive on these things. This requires this. We need glycolysis. Glycolysis needs to be activated in the brain. We need it in the brain. So, I'm just gonna put it here. Is important. So there's different organs where glycolysis is important. I'm just going to mention them a bit. So we have the brain, where glycolysis is important. And it's important because, again, it can't use fat as fuel. So cannot utilize fats as fuels. Is also important in red blood cells. Okay, so glycolysis is important in your red blood cells because, as you know, mature red blood cells do not have mitochondria, and mitochondria is needed for the TCA cycle, oxidative phosphorylation. I'll talk to you about that after. And glycolysis is also important in your skeletal muscle. Okay, during intense exercise. Okay, so when you're exercising a lot, you need that energy. So that's what I want to talk to you about, and things which can block these things. So I'm kind of going in extra detail now. So if you guys want to stop the video now, that's completely fine by me. But what I want to talk to you about now, guys, is something called an allosteric regulation, which allows regulation of certain enzymes. So if I just tell you, for example, when you're going from fructose 6-phosphate to fructose 1,6-bisphosphate, that happens by your famous enzyme phosphofructokinase. Okay, simple as that. But there can be certain allosteric regulators. So, for example, there can be ATP. Okay, and ATP will block this pathway. There can be also citrate. Citrate will block this pathway. And there can be, however, on the other hand, there can be things like, let's say, I'm just going to put a squiggly line just to show you this. They can be adenosine monophosphate. And these things stimulate fructose 6-phosphate to go into fructose 1,6-bisphosphate. So these are allosteric regulators. They can be allosteric stimulators or allosteric inhibitors to prevent fructose 6-phosphate to go into fructose 1,6-bisphosphate, such as ATP and citrate. But they can also be allosteric stimulators, allowing this process to take place. And, if you notice other things as well, which I'll talk to you again about a bit later on in my lecture series, you have reversible reactions here, reversible reactions everywhere. Okay, so you have one here, you have one there, you have one everywhere. Okay, there's only 3 irreversible reactions in the whole glycolysis cycle. Okay, so only 3 irreversible reactions. Okay, and these 3 irreversible reactions require an enzyme. Okay, so we need an enzyme to actually overcome these irreversible reactions if we want to go reverse glycolysis. Meaning, if we want to go from our pyruvate back into our glucose. If we want to do that, we have to overcome those 3 irreversible reactions, i.e. use different enzymes, but use the same enzymes to go in the other way for the same ones, for the reversible ones. So yeah, guys, so I hope you enjoyed this video. This is kind of the, I don't know how how much detail as well, you know, any lecturers or anything like that, how they want you to get into it. But if you have any more further questions on my glycolysis, you know, on the glycolysis cycle, feel free, guys. Don't be scared. Leave your comments down below. I'll definitely answer them. And I'll obviously make you understand this a bit more further if you didn't understand my lecture already. But yeah, guys, that's the glycolysis cycle. I hope you enjoy the glycolysis cycle now. Just a quick word of notice. This is a cycle which, you know, you can't cram. This is something you have to understand. You know, I've been doing this from memory now, and this is just something you have to know. You know, you can't, you know, not know it. Okay, so I hope you enjoyed this video, guys. If you, again, practice makes perfect. So try and drawing it out yourself from memory. You know, try, you know, putting everything in, check it again. That's what I've been doing, and, you know, it worked amazingly. So you guys can do that as well. Hopefully you'll do good.